Good morning, everyone. Welcome to our webinar on RNAscope data interpretation. My name is Rob Monroe, and I'm the Chief Medical Officer of Advanced Cell Diagnostics. I'm also a board-certified pathologist trained in anatomic and clinical pathology. This morning, we're going to be talking about data interpretation for the RNAscope RNA in situ hybridization assay. The agenda we'll be going over is shown on the first slide. We'll begin with a brief overview of the RNAscope technology, talk about a few of the common RNAscope applications, and then dive into a review of RNAscope staining and scoring, including a comparison with immunohistochemistry, an overview of some of the definitions that we use for RNAscope signals. We'll talk about the sensitivity and limit of detection of the assay. Then we'll move on to a review of the manual scoring systems that can be used for RNAscope, including qualitative, semi-quantitative, and H scoring. We'll also cover image analysis and quantitative scoring before finishing up with an overview of the quality control procedures involved in performing and interpreting RNAscope. With that, I will begin with a brief overview of the RNAscope technology. As many of you know, there's been an unmet need in research and tissue diagnostics over the last 3 decades for a reliable RNA detection method to complement the very well-established and highly utilized FISH and IHC techniques that have been used for the detection of DNA and proteins, respectively. There was no sensitive or robust technology for the reliable in situ detection of RNA prior to the development and commercial launch of RNAscope in 2010. So what are the fundamentals of the RNAscope platform that allow for this sensitivity and robustness in performance relative to some of the prior techniques used for RNA in situ hybridization? To start with, RNAscope is an assay that can be performed on a variety of tissue types, including formalin-fixed paraffin-embedded tissue, which is critical as it's one of the most common tissue types in both research and development as well as clinical settings. The assay can also be performed on frozen tissues and other tissues, so it's not limited to FFPE, but it certainly has been optimized for FFPE. RNAscope utilizes a unique probe design for each target indicated in the purple and orange, and this relies on the so-called double Z probe strategy depicted here with the 2 double Z probes lined up along the target. And this strategy ensures maximum specificity of the assay. The probe design, in combination with the amplification steps, which I'll get into in the next couple of slides, provide for an approximate 400-fold amplification of the target, concurrent suppression of any non-specific background staining. The sensitivity of the assay allows for detection of single RNA molecules in single cells as punctate dots in their morphologic context. So you can see here the representation of a microscopic field with individual cells and punctate dots within them representing the individual RNAscope signals. These punctate dots can be easily visualized with standard microscopes, both light microscopes as well as fluorescent microscopes, and then quantified either manually through standard microscopic assessment or with software programs that can actually count the dots, and I'll go over a couple of those applications a little bit later in the talk. Overall, the technology is broadly applicable for the detection of almost any biomarker in any species. Returning to the probe design, technology is based on the use of V-shaped oligonucleotides composed of a lower region of approximately 25 base pairs connected through a spacer to an upper tail region. when a pair of double Z probes, or Z probes rather, hybridize next to each other along a target sequence, they identify a target binding site of approximately 50 base pairs. This hybridization to this target site creates a so-called pre-amplifier binding site at the top of the double Z pair, which allows for the subsequent building of an amplification tree and amplification of the signal. The target probe set for a given biomarker consists of approximately 20 double Z probe pairs spanning a target region of about 1,000 base pairs, which is depicted in the right-hand panel of this slide. So you can see each of the double Z pairs lining up along the target biomarker sequence. The target probe is designed using very well-established bioinformatic tools to provide for an assay which we guarantee will be single target specific. This double Z probe design is really the foundation for the highly sensitive and specific messenger RNA detection that is one of the hallmarks of the technology. The steps following the hybridization of the double Z probes along the target sequence provide for signal amplification and further specificity. Pre-amplifier molecules shaped like an L, indicated with the laser pointer here, will only bind if they encounter a pair of these adjacent double Z probes, which have the so-called pre-amplifier binding site. In subsequent amplification steps, the hockey stick-shaped amplifiers and circular label probes, indicated in green, bind to the pre-amplifier and amplifier targets respectively, leading to an approximate 400-fold amplification per double Z pair. If a Z probe binds to a non-specific target, the pre-amplifier will not bind. And no signal will be generated. Overall, for a 1 kilobase target region, the amplification strategy has the potential to recruit approximately 8,000 labeled molecules, which is equivalent to the product of 20 of these double Z pairs multiplied by 20 amplifier per pair with 20 labels per amplifier. The double Z strategy derives its robustness and reproducibility for the detection of RNA molecules through the redundancy of the double Z probes. Although most RNAscope target probes utilize approximately 20 double Z pairs, hybridization of only 3 of the 20 total pairs is required for detection of a single RNA molecule. So really, what's required is only hybridization of about 3 of a total of 20 double Z pairs to generate a target. This redundancy allows for the detection of fragmented targets in the context of FFPE tissues, which can have breaks in the RNA, as well as bound proteins indicated here. As a result of these features built into the RNAscope technology, RNAscope is about 100 times more sensitive relative to traditional RNA in situ hybridization methods, which utilize single joxygenin-labeled probes, as shown here in a direct comparison of RNAscope on the left versus traditional ISH on the right for detection of simian immunodeficiency virus in lymph node tissue. While the details of the performance of the RNAscope assay, the technical details are beyond the scope of this webinar, RNAscope can be performed both manually as well as with the help of automated equipment. ACD is designed, on the automation side, ACD has designed both specific kits and optimized protocols for Ventana as well as Leica automated staining platforms indicated in the slide. Presently, automation is only available on the Ventana Discovery, as well as the Leica Bond RX, both research instruments from these companies. But in early 2016, select RNAscope assays will be available on Leica's clinical platform, which is known as the Bond 3, for use in clinical labs as laboratory-developed tests. Overall, automation not only has the potential to save significant hands-on time for researchers and laboratorians, but it also provides for greater reproducibility in assay performance, both from day to day, batch to batch, as well as operator to operator. So this really improves on the reliability and robustness of the assay. Before diving into the details of RNAscope scoring, I thought it would be instructive to review a few of these many applications RNAscope has described in some of the many publications that have utilized the RNAscope technology. As a result of the high sensitivity and specificity for RNA detection that I've described through this methodology, this RNAscope methodology, RNAscope has been widely adopted and through August of this year has been used in over 540 peer-reviewed publications spanning most all of the top journals, including the New England Journal of Medicine, Cell, Science, Nature, and many others. RNAscope assays are applicable to a wide variety of fields, publications spanning cancer biology, immuno-oncology, stem cell biology, infectious disease, inflammation, developmental biology, neuroscience, and many other fields. So it's really a technique that lends itself to investigations in the spectrum of R&D as well as diagnostics. Getting into a few applications prior to a review of the staining and scoring, one of the most useful applications for the technology is for the detection of intratumoral heterogeneity for the presence of specific biomarkers and genetic changes. In this prostate cancer study, dual RNAscope staining for the ERG and ETV1 targets, ERG shown in brown and ETV1 shown in red, the red chromogen. Both of these biomarkers are overexpressed in prostate cancer as a result of gene fusions. And in this study, you can see the differential and mutually exclusive expression of the two biomarkers in different parts of the tumor. So in the left-hand panel, you can see expression of ERG, but not ETV1. In the right-hand panel, you see a beautiful expression of ETV1 in the absence of ERG overexpression. This combination of molecular detection and morphological context that can be provided by RNAscope is not possible with traditional grind and bind NGS or PCR assays, which destroy morphology, as well as with standard IHC assays, which don't provide the genomic information that RNAscope can provide. It goes without saying that immuno-oncology is one of the most exciting areas of research at the moment. RNAscope has shown to be a great tool for the identification of specific cell types of interest within tumors, expressing various immuno-oncology markers such as PD-L1 and other cell surface receptors and cytokines. In this example of a dual RNAscope assay for PD-L1 and CD8, in a case of a non-small cell lung carcinoma, the green signals represent PD-L1 expression by lung cancer cells, highlighted by the green arrows. So the green sometimes shows up as, it may show up as blue on your screen. Extensive infiltration of CD8 positive cytotoxic T cells throughout the tumor is indicated by the red signal. So you can see the red chromogenic signal. And these are highlighted by the pink arrows. Occasional co-expression of PD-L1 and infiltrating CD8 positive CTLs is noted by the yellow areas, by the yellow arrows rather, indicated in this cell here, where you see both red as well as green punctate dots. RNAscope can be employed in both chromogenic assays, as shown in the previous two examples, as well as fluorescent assays, as shown here. In this example of CTC detection in a breast cancer patient, RNAscope target probes for CD45 positive bone marrow derived cells and for pan-keratin positive circulating tumor cells were used in a duplex assay. CD45 positive cells show punctate red dots, whereas the CTCs are highlighted by clusters and clumps of green dots. So you really see the epithelial cells stand out in this peripheral blood representing a CTC context of a blood sample. Up to 4 different RNA markers can be assayed in these fluorescent assays. ACD now has a kit for allowing this type of analysis. In addition, these fluorescent assays can be adapted to allow for detection of chemistry in one channel, so up to 3 RNA biomarkers can be assayed along with a single protein in a combined IHC RNAscope assay. Particularly in the field of head and neck cancers in humans, RNAscope assays for human papillomavirus are proving to be very valuable as a clinical diagnostic test. These tests are important for the characterization of squamous cell carcinomas of the head and neck area, particularly a subtype known as oropharyngeal carcinoma. Numerous studies have shown a difference in the behavior and aggressiveness of HPV-related oropharyngeal carcinomas versus non-HPV-related tumors, which are typically thought to be related to smoking and alcohol exposure. Because the HPV-related cancers are less aggressive, the clinicians like to know this information so that they can provide potentially less intensive treatment regimens for the patients, as well as provide the patients feedback that their tumors are likely to behave better. In other words, that they'll have a better prognosis. The final application that I'll briefly mention prior to getting into the data interpretation portion of the webinar is the application of companion diagnostics. So RNAscope is being used by several biopharma companies in their companion diagnostic programs, including Merrimack Pharmaceuticals. And in this slide here, I wanted to illustrate the strategy that Merrimack is using to stratify a group of lung cancer patients that they're considering for treatment with the MM121 treatment, which is an anti-HER3 monoclonal antibody. They're using the RNAscope assay for a biomarker called heregulin, or NRG1, to stratify patients for treatment with this anti-HER3 monoclonal antibody. In patients that have expression of heregulin, as shown in the bottom microscopic panel, you can see these punctate dots through the RNAs that are identified with the RNAscope assays, suggesting or documenting expression of the heregulin RNA molecules. These patients appear to respond more favorably to treatment with the anti-HER3 monoclonal antibody, and this is a strategy that's being used again by other biopharmaceutical companies in the development of some of their drugs as an alternative to other assays such as immunohistochemistry or PCR-based methodologies. So moving on to RNAscope staining. Let's start with a quick comparison of RNAscope and immunohistochemistry. Both assays can be performed in either chromogenic or fluorescent formats. Both are also compatible with FFPE tissues. It can also be applied to tissues fixed under other conditions, whether that be alcohols or even fresh frozen tissues. Regarding the signal appearance, IHC assays tend to produce a diffuse signal in the region of the cell in which that protein biomarker is located, while RNAscope assays produce dots and clusters, which we'll go over the definitions of in a minute. The subcellular localization of staining for immunohistochemistry varies with the biomarker. For RNAscope, the signal is typically in both the nucleus and the cytoplasm, reflecting detection of transcription in the nucleus and export of transcripts into the cytoplasm for translation. Last, both IHC and RNAscope work in almost any cell type. Some of the common immunohistochemistry staining patterns are shown and listed here, including nuclear, cytoplasmic, nuclear and cytoplasmic, membrane and cytoplasmic, and even other subcellular compartmental staining, typical membrane staining, for example, and on down the line, indicative of expression of proteins in specific organelles, for example. In comparison to the diffuse staining pattern of most immunohistochemistry assays, such as this example of a P16 stain on the right, RNAscope assays show dots and clusters in nuclear and cytoplasmic compartments. So again, in the immunohistochemistry assay, you can see diffuse staining of the protein in both the cytoplasmic as well as the nuclear compartments, whereas in the RNAscope assay, you can visualize both dots and clusters overlying the nucleus as well as surrounding the nucleus in the cytoplasmic compartment. So what are some of the definitions for the terminology used for these RNAscope assays? What does a dot mean? What is the significance of dot size? What is the difference between a dot and a cluster? And what is the sensitivity of RNAscope versus IHC? And over the next couple of slides, I will address these questions in an attempt to provide the basic vocabulary for description of RNAscope assay, staining, and data interpretation. So RNA or messenger RNA molecules are detected as punctate brown dots in the RNAscope assay. The difference between a dot and a cluster is highlighted in this particular image. Again, single messenger RNA molecules are detected as punctate brown dots, while larger dots or clusters result from overlapping signals from multiple messenger RNA molecules. In general, variation in dot intensity can be related to differences in the number of the double Z probes that we talked about that are bound to a target molecule. So in this image, these two arrows on the left are highlighting likely individual messenger RNA molecules, whereas the two red arrows on the right are indicating clusters representing multiple messenger RNA molecules and the signals from those coming together to form a cluster. Similar patterns of dots and clusters are seen in fluorescent versions of the assay. Again, you can see in this next to this arrow a likely cluster of green dots relative to a single dot here representative of a likely single messenger RNA transcript. In general, RNA expression for a given biomarker is related to dot number rather than dot intensity. And the dot intensity itself is typically related to the number of double Z probes bound to a target molecule in signal amplification steps. So, in other words, for two different biomarkers, if the number of dots observed in those assays is similar, they're likely to have similar levels of mRNA expression, even if the dots in for one biomarker appear to be slightly more intense biomarker. The detection limit for RNAscope is typically below the detection limit for immunohistochemistry. In this example, comparing RNAscope and immunohistochemistry detection of biomarker CMET, you can see that in the low-expressing CMET cell line in the top row, there's no detectable signal in the immunohistochemistry assay. In this upper right-hand panel, no protein or immunohistochemistry signal is seen. However, there are clearly RNAscope dots well above and beyond the background negative control background in the RNAscope panel, shown here and highlighted with the red box. This really demonstrates the ability of RNAscope to pick up low level of expression not typically seen by immunohistochemistry. In comparison to real-time PCR or quantitative PCR, RNAscope shows similar sensitivity and limit of detection in experiments looking at expression of the HER2 biomarker in the spectrum of human breast cancer tissues in the left hand panel. The number of dots per cell was highly correlated with transcript levels as determined by real-time PCR. So again, in the y-axis, you can see the RT-PCR compared along the x-axis to RNAscope dots per cell, and you see a nearly linear line with a similar lower limit of detection. So it's really a quantitative, has the potential to be a quantitative assay similar to real-time PCR assays. Moving on to manual scoring. In general, manual scoring can be performed at an approximate 20x magnification. If high RNA expression is evident for a given biomarker in a given RNAscope assay, that expression can typically be seen at 10x magnification. For lower expressors, examination at 40x magnification typically is required, both to observe the dots to make sure that they're there or to determine whether they're there or not, as well as to potentially count the dots and assess these low RNA expressors. Moving on to scoring, there are a number of ways to score RNAscope assays, ranging from qualitative to semi-quantitative, H scoring and image analysis, which we'll cover. For some assays, particularly those looking at markers that are highly expressed when present, simple qualitative scoring can be appropriate. The panel in this slide shows clearly positive expression of albumin in a case of hepatocellular carcinoma on the left, in comparison to a clearly negative expression of bacterial gene performed an RNAscope assay on the same tissue on the right. Similarly, in this panel, you can see obvious positive expression for cytomegalovirus, in this case, in a case of CMV chorioamnionitis. Again, quantitative scoring isn't necessary, especially for some of the clinical diagnostic applications where the presence or absence of the biomarker is the most important issue rather than precise quantitation. Semi-quantitative scoring systems are common for IHC assays. One of the more commonly used systems is shown here with a scoring range of 0 to 3+, with the corresponding staining intensities. So this is a very typical scoring schema for many labs using immunohistochemistry, ranging from a score 0 to a score 3+, representing the spectrum from negative through weak, moderate, and strong staining. Similar semi-quantitative scoring systems have been developed and are commonly used for RNAscope assessment. The scoring scheme shown in this slide is what is routinely used by ACD and its pharma assay services group. It spans a scoring range of 0 to 4, corresponding to the number of dots per cell. A case with a score 0 has essentially no staining, and less than 1 dot per every 10 cells. Moving down the scale, a score of 1 corresponds to 1 to 3 dots per cell, a score of 2, 4 to 9 dots per cell, and so forth, all the way through a score of 4, representative of 15 dots per cell, with more than 10% of those dots arranged in so-called clusters that we defined earlier. This is just an example of a semi-quantitative scoring system that can be used and that we often use internally here. Other scoring systems are acceptable. For example, More granularity is required to identify a larger number of subsets of cells with different staining patterns. This type of schema can be expanded to include additional categories, or it can be compressed to allow for the binning of the cells in a smaller number of categories. A representative example of a semi-quantitative score of 0 for this scoring system that we just went over is shown in this panel with clearly absent dots in this photomicrograph. These panels show a score of 1, with most cells containing between 1 and 3 dots, so the dots are a little bit difficult to visualize because of the darker hematoxylin stain on the left, but there are approximately one dot per cell that are visualized. A little bit easier to see in the cells in the right-hand panel, where you clearly see that it's in the range of 1 to 2 dots per cell. These panels show a score of 2, with most cells containing in the range of 4 to 9 dots. Very few of these dots are present in clusters. So this is a representation of score 2. These panels show a score of 3, with most cells containing 10 to 15 dots. Less than 10% of the dots in these cells being present in clusters. And finally, these panels show a score of 4, with most cells containing greater than 15 dots. Many of the dots in these cells being present in clusters. So moving on to H scoring. As for IHC, H scoring is also compatible with RNAscope. For those of you who are not familiar with H scoring, it's basically a method of assessing the extent of staining for a given biomarker in a tissue, incorporating both staining intensity as well as the percentage of cells with given staining intensities. So for IHC, the H score is obtained by the formula, again using a scoring category of 0 to 3, the most common H scoring schema. Each score is obtained with this formula that I won't go through, but it basically involves determining percentage of cell staining in each of these categories to obtain a score in the range from 0 to 300. For RNAscope, staining categories are determined, again, by the number of dots per cell rather than staining intensity, but H scores can otherwise be derived in a very similar manner. Moving on to image analysis, which is a way to provide for more quantitative scoring than some of the methods that we've discussed so far. For quantitative assessment of RNAscope studies, ACD has employed image analysis software in many of its internal studies, and these image analysis software, this image analysis software is also available for other researchers and laboratorians if desired. There's multiple different software applications available for this type of dot counting that's required for assays like RNAscope as well as DNA FISH. ACD and its internal pharma assay services group commonly use the two software applications highlighted in this slide. The first is SPOTSTUDIO, which is a custom designed algorithm developed by ACD in partnership with Definiens that enables quantitative analysis of chromogenic single-color RNAscope assays. Again, typically these software applications are utilized on whole slide images that are obtained with digital pathology whole slide scanning systems. HALO is a second software application offered by Indica Labs that provides similar quantitative analysis for not only singleplex assays, but also more complex duplex and multiplex RNAscope assays. So this is the software application that we use more commonly internally because of its ability to look at multiple colors. An example of SPOTSTUDIO image analysis, in this case for the biomarker HER2, breast carcinoma is demonstrated in this slide. The software identifies cell boundaries shown with this sort of chicken wire lattice work through the approximation of average distances between the cell nuclei. The algorithm then counts dots within each cell as defined by these cell boundaries and bins the cells by color according to the number of dots per cell. So the analysis shows blue cells, approximately 10% blue cells that have 0 to 1 dot per cell, and so the blue cells are highlighted by the blue boundaries, similar for green, where approximately 30% of the cells contain between 1 and 3 dots and so forth. The HALO software can perform similar analyses to SPOTSTUDIO on singleplex assays, but also allows for more complex duplex analyses. So in this case, you can see the red chromogen, as well as a little bit harder to see, but there's both red as well as brown chromogen. So it allows for quantitation of duplex assays. In HALO's software, H-scoring is also a feature. This quantitative H-score analysis is highlighted in this slide. In this type of analysis, each cell is assigned to a bin, similar to what I described before, that's based on the number of dots per cell. Clusters are divided by the typical probe signal area, calculated the dot number for the clusters. So in other words, the size of that cluster is determined by the software, and then based on that size, an average number of single mRNA molecules are determined from through the software algorithm. An H-score calculation can then be performed on the entire tissue, the entire region of interest, to assign a single score to a selected area based on the average target expression in this tissue. In this case, the H-score for the given biomarker was 147 out of a possible 400. So moving on to quality control for RNAscope results, and this is a very important feature of the assay that allows for more accurate interpretation and more definitive conclusions to be made for researchers and laboratorians performing the RNAscope assay. So for proper interpretation, RNAscope assays should always be performed with concurrent positive and negative controls. Positive controls are typically housekeeping genes expressed at similar levels in most tissues. The common positive controls utilized in RNAscope assays include a marker called UBC, another marker called Pol2RA, as well as a third marker called PPID. Their use is really analogous to the use of genes like beta-actin and GAPDH in RT-PCR and northern blot experiments. The purpose of the positive controls is to demonstrate the integrity of RNA for RNAscope testing. So in general, if signal is present, in other words, if there's chromogenic dots or dots are present in the assay, performed with the positive control probe, tissue is suitable for RNAscope analysis with a target probe, whereas if the signal is absent, the tissue is generally not suitable for RNAscope. Indicates that the RNA in that particular tissue specimen is likely to be degraded or compromised in some way, such that proceeding with that tissue for a target probe assessment is not worthwhile. The negative control typically used for RNAscope assays is a probe targeting a bacterial gene called DAP-B, which is from a soil bacterium that's very unlikely to have any sequence homology to any of the sequences that are being probed by our various investigators using the technology. In general, If a signal is present using this negative control probe, tissue is not suitable for RNAscope and indicates that there's some issue with either the performance of the assay or the tissue itself. The signal is absent, and that indicates that the tissue is suitable for RNAscope. Depending on the target, different positive control probes are recommended, as highlighted in this chart. For example, the housekeeping gene UBC, which has a medium to high expression level, should be used with high expression targets or high expressing targets but not low expressing targets. Because it's highly expressed, the UBC signal may be picked up even in the presence of significant RNA degradation, and therefore can lead to false negative target results, particularly for biomarkers with lower levels of expression. PPIB has a more moderate level of expression and is recommended for use as the positive control for most tissues. Its expression is low enough to provide a rigorous control for RNA quality of a sample. And in the vast majority of studies, if the PPIB signal is present, then any target probe will detect your target RNA. Finally, the Pol2RA housekeeping gene in RNAscope assay has a low expression level and is recommended to use with low expression targets. Well, some of the staining patterns of the control probes is highlighted here. UBC again is the highest, has the highest level of expression of the housekeeping genes that are typically used in RNAscope assays. Again, PPIB is generally recommended for the positive control probe. For most studies, as it has a low to moderate level of expression relative to UBC, such that if signal is seen, the RNA is typically suitable for detection of most any target. So moving on to QC methodology, setting up an experiment to test methodology in a laboratory prior to running a target probe on target tissues of interest. So prior to running an RNAscope experiment with target probes, the technique in the laboratory should be checked and samples should be qualified with the appropriate positive and negative controls that we just described. In the first step, slides prepared from cell lines can be tested with the appropriate positive and negative controls. In this case, DAP-B as well as PPIB for the negative and PPIB for the positive. If the PPIB signal is greater than 2 dots per cell, as indicated here in the lower panel where you can see the brown chromogenic signal, and the DAP-B signal similarly shows less than 1 dot per cell, so you can see in the upper panel here, tissue essentially has no brown staining. The RNA quality under these, with these results, again, the PPIB greater than 2 dots per cell and DAP-B less than 1 dot per cell, that would indicate that that assay is working well on those control tissues in that laboratory. In the next step, moving on again from potentially cell lines and control tissues to a sample tissue, the same set of positive and negative control probes are used to test the sample. Again, if the PPIB shown in the lower panel shows greater than 2 dots per cell, and the DAP-B, the negative control, shows less than 1 dot per cell, then that sample can then be moved on to assess with the target probe of interest. If the target probe back 1 second. So in this case, as I mentioned, if the PPIB is greater than 2 and the DAP-B is less than 1, and that tissue is essentially ready to go on for experimentation with any number of target probes for that particular tissue specimen. The following example case provides a step-by-step review of the QC process. In step 1, A technique check with the cell line controls is performed and shows abundant UBC staining in the positive control panel on the left and absent DAP-B staining in the negative control on the right. QC clearly passes, showing that the technique is working in that particular laboratory. In step 2 of the example case, the tissue sample is checked for RNA quality with application of positive and negative control probes. In this case, again, it looks very similar to the previous slide. UBC is clearly positive, so you can see the brown chromogenic dots in this tissue, while the DAP-B is clearly negative, supporting sufficient RNA quality for this particular tissue specimen to proceed with subsequent RNAscope testing. In step 3 of the QC example case, tissue sample is tested with several different target probes now that it's been qualified as having intact RNA without significant background staining. And in this case, the target tissue was stained with several probes for different HPV subtypes, including HPV-16, HPV-18, HPV-31, and 33, performed in a duplex assay, as well as HPV-6 and 11, again, performed in tandem. You can see that HPV-16 is strongly positive, whereas the other HPV subtypes are negative, indicating that this tissue is positive for infection by HPV-16. The fourth optional step can also be performed and is recommended if known positive and negative control cell lines or tissues are available, similar to the use of positive control tissues for immunohistochemistry. In this case, a cell line known to be positive for HPV-16 and a cell line known to be negative for HPV-16 are employed to demonstrate the specificity of HPV-16 probe staining in the target tissue. So HeLa cells are known to be negative for HPV-16, whereas Casti cells are known to be positive for HPV-16. So once the technique, the RNAscope technique is working reproducibly, labs can skip the first step that I highlighted in the QC procedure, which is the technical check, and then run steps 2 and 3, which is the tissue sample check with the control probes, followed by the tissue testing with the test probes. Those subsequent steps 2 and 3 can be run concurrently. In this RNAscope experiment shown for assessment of EBV in a target tissue of interest, this strategy was done where, since the assay was working in the lab proceeded immediately to testing the target tissue with positive control probe, the negative control probe, as well as the target probe, which is EBV in this case, all concurrently in order to save time. And this is really the most efficient way to use the assay once it's up and running in the lab. In this experiment, you can see that obviously the RNA is intact, as indicated by the strong staining with the housekeeping positive control probe, PPID. No significant background staining, as indicated by the negative appearance of the panel stained for DAP-B. Then you have a very robust, strong signal for EBV. In this case, the dots are so numerous, they essentially coalesce to create what almost looks like an immunohistochemistry, a diffuse immunohistochemistry staining pattern for the EBV. This is characteristic of many viral infections where the virus really takes over control of the transcription machinery of the cell and produces hundreds, if not thousands, of messenger RNA molecules, which are, in this case, detected by the RNAscope assay. So once RNAscope testing becomes routine for a lab, particularly if automation is available, the sky is really the limit. And I think I'll end on that note for the data interpretation portion of this webinar. And just finish up with the following slide. As you know, with all the numerous applications possible, only a few of which we've discussed at the start of the webinar, the RNAscope technology can really be applied to any target in any species to support a variety of exciting research and clinical diagnostic needs. Some of the important features of interpreting the data and scoring were covered in this webinar, but this is really only an introduction to help you get going with the assay in your own laboratory. And with that, we will open up the webinar for questions. Thank you very much. The first question that was submitted was, how do you derive an H-score from RNAscope spot counts? That's a great question. And so the image analysis software makes the H-score easier to calculate, but it can also be done manually. Basically what's done is the percentage of cells falling into different different numbers of, having different numbers of dots is determined. So for example, the number of cells with no dots is determined and converted to a percentage. Then the number of cells with 1 to 3 dots is determined and converted to a percentage, and so forth for the number of categories, for the number of staining categories. And then each of those percentages is multiplied out to derive that H-score. And I'm happy to provide an example if you or your email on how to do that for your particular biomarker. The second question is a follow-up to that. First question, does the IHC H-score correlate with the RNAscope H-score? That would really have to be determined on a case-by-case basis. In general, what we found is that protein expression correlates quite well for many biomarkers with mRNA expression that's assessed by RNAscope. But in some cases, the RNA may be more or less stable than the protein, and so there can be differences in what's observed when you're comparing protein levels for a given biomarker to mRNA levels. And so again, it's a biomarker by biomarker scenario that needs to be considered. For some, it'll be comparable. For some, there may be some differences. The next question is, are non-coding RNA dots only in nuclei? That's a very good question, and it depends on the type of non-coding RNA. From my understanding of the different types of non-coding RNA, there are some that can also end up in the cytoplasm and, for example, be involved in structural, have structural roles outside the nucleus. And so those dots for those particular non-coding RNAs could be both in the nucleus as they're being transcribed and then in the cytoplasm as they're exported. So in general, that non-coding RNAs can be seen as dots in both compartments. The size limits of the probes, that's another great question. We have a standard technology that aims to design probes to approximately 1,000 base pair target, and that's how we end up. If we have the sequence available, we'll generally try to design a probe that spans 1,000 base pair target with about 20 double Z pairs. But that's by no means required. We can go down to a couple hundred base pairs with as few as 8 to 10 or 8 to 12 double Z pairs. And so it really depends on the level of expression of that messenger RNA molecule. In general, for the lower expressors, it's nice to have a longer sequence that's targeted, whereas some of the high expressors you can get away targeting a much shorter region of DNA. The next question is, can I replace DAPI counterstaining with other staining methods, such as NISL? I don't see why not. The DAPI, in the context of the fluorescent assays, is just a marker for the nuclei, and if that channel is available, or if you'd like to replace that particular channel or stain the nucleus with a different color, that's certainly possible. DAPI in our hands is generally used mainly for the identification of nuclei and being able to determine where one cell starts and one cell ends. The next question is, scoring tumors with heterogeneous expression in different regions and different scores. That's a great question. As we know from IHC, there can be differences in expression of protein in different areas of the tumor, with some having higher levels of expression, some lower, and some none. I showed an example of ERG expression as well as ETV1 expression in a prostate cancer that varied in different parts of that particular prostate cancer. And so again, it's dependent on the biomarker being assessed, and certainly can be done, and it's been seen in many of our investigations, particularly in tumor biology. And so the important thing to determine is whether technical aspects of the procedure were performed and that the tissue is uniformly stained, for example, for the positive control probe, so that and then actually determine whether differences in the target probe expression across the tissue is related to true differences in expression or relative to a potential issue with staining RNA quality, staining quality or RNA degradation across the tissue. So just something to keep in mind. But in general, differences in staining can be quite common, particularly in oncology. Next question is basically asking whether fixation time has an impact on the assay, particularly for researchers who may have to purchase tissues who don't have control over the fixation time. So in general, fixation can be an issue if the tissues are either underfixed or overfixed. In the case of underfixation, the RNA can be degraded if there's RNases that are still hanging around, or if the RNA is not stabilized in a way that allows for adequate preservation. On the other hand, if tissues are overfixed, then the number of breaks in the RNA can be such that the assay is not effective in identifying those RNAs because of the fragmentation. So the sweet spot is somewhere between 12 and 48 hours. Generally, the lengths of fixation are recognized by most of the vendors as an important variable, not only for RNAscope and RNA ISH, but also for immunohistochemistry performance. So I think that there is an attempt to control for that as much as possible by the vendors of these tissues. But it is an issue, and if you can obtain that information from the vendor and try to optimize the selection of tissues in a window of fixation that's most compatible with RNAscope, then that would be preferable. But in general, the flexibility of the assay for fixation is quite broad, and tissues fixed, again, as long as they've been fixed for several hours up to a couple of days, RNAscope generally works quite well. The next question is, if there's a negative control signal on a significant number of cells, how do we change our tissue preparation to make it acceptable? That's a great question, and goes back to the QC technique in optimizing the assay in a given laboratory for a given tissue. Some tissues are notorious for having a higher amount of background signal with the negative control probe. So for example, liver and spleen are tissues that can be difficult to work with, and often among spectrum, at least among human samples, give the higher levels of background. So what we generally recommend is working with our customer support team to work on the pre-treatment conditions. So those can involve fixation times, which we just talked about. They can also involve the time that protease is used for digestion of the cells prior to the hybridization steps. So protease pre-treatment, we can either increase that or decrease that depending on the background that you're observing, and we can also adjust some of the other pre-treatment conditions, including heating. And so that's something where I would urge you to reach out to our customer support team. I just put up the contact information on the screen, and we can help you work through that issue with your negative control probe having a higher signal than you'd like. The final question, and this will be the last question that we have time for today. The final question is integrated density function of image J suitable for relative quantitative analysis using fluorescent probes? If we have the same settings for all pictures and set thresholds, so it does not take the background. That's quite a technical question. If you would submit that to the support email address shown on the screen, that would be great, and we'll see what we can do, since that's specific to your specific software application, but we should be able to come up with an answer for you on that. With that, I'd like to wrap up the webinar today. Thank you very much for your attention and participation. Please, again, reach out to our ACG support team for any technical questions, and we look forward to helping you optimize your assays as well as interpret your RNAscope data going forward. Thank you again, and have a great day.